Our next speaker is Dr. Rabi Tawil from the University of Rochester, and he will be telling you about actual clinical trials, which ones are going on or pending, and maybe a little glimpse into the future beyond that.
Thank you, June, and thank you, Dan, for inviting me. And it's nice to see some familiar faces out there. June's instructions to us was to make it as simple as possible and make it like a TED Talk. I was really, I felt very important that I'm giving a TED Talk, but it's, but this is a very sophisticated audience, and maybe this was a little bit too simple, and again, some of that you've already seen, but I think it's important to again look at it, and maybe this is going to be in a form of a summary of what we've talked about. And so, this is kind of very simple. DUX4 is not supposed to be expressed in muscle. In FSH, it is expressed in muscle. And DUX4 is translated into protein, which is toxic to muscle. And this leads to weak and atrophic muscles, as you see on the right side, and the whole purpose of treatment is to reverse that. And so that can be done in several ways. And again, this was presented, Peter Jones talked about this a little bit. And there are many ways, many places where we can interfere as far as the effect of DUX4. Obviously, the most effective is getting it at the root of the problem, which is DUX4. So this would be the most effective way of treating FSH if you can do that. And there are ways of doing that. And again, I think there's the question that I always hear is how many years until we have a trial? And we're all very optimistic. And I can tell you that in another muscular dystrophy, myotonic dystrophy, which my colleague in Rochester did a lot of work on, he can tell you that from concept to clinical trial for the use of antisense oligonucleotide in myotonic dystrophy, it took 2 years. So not that this is going to be 2 years for FSH, but what I'm saying is that the technologies there has been applied in other diseases, and so the trajectory from lab to clinic is bound to be quicker. Although, again, as Scott was saying, I mean, it's never linear. There are always problems, but we have a lot of companies interested, a lot of very good researchers, and so this is not a. I think this is a very realistic prospect in the near future. So what happens, this is when DUX4 is not, is leaking when it shouldn't be, when it should be turned off. Intervening at DUX4, you can either turn it into just a drip, and improve function perhaps, or at least slow down progression. But ideally, what you want is to turn it off completely. And again, there are ways of doing that. We don't know how close we're going to get to a faucet that's completely turned off, but certainly there are ways of blocking DUX4, and it's a matter of taking it from the lab to the clinic. A less effective way, but nevertheless important way, is to look at what we call the downstream effect of DUX4. What is it that DUX4 does that leads to muscle weakness and muscle atrophy? And there are many things that we know about. Some of these were listed before in the table that Peter showed. Inflammation, oxidative stress, problems with muscle regeneration, apoptosis, which is programmed cell death, and muscle atrophy, which is the consequence of all of these things. And so one of the things we don't know about is which of these multiple things that we know is going on in muscle is the fact that's contributing the most to the muscle atrophy and weakness, and that we don't know. And so, different people. The group in France took a stab at trying to address oxidative stress, and that study is done. There were some laboratory evidence that there was some improvement in oxidative stress, but it was not clearly, it did not clearly result in clinical improvement in those patients. Currently, there are two drug companies that are working on, one is working on targeting inflammation, and the other one is working on targeting muscle atrophy. And again, these are not necessarily targeted treatments, but important nonetheless, because if you do get responses from these interventions, as was discussed before, these could be additive treatments to actually knocking down DUX4. If DUX4 is either not completely knocked down or knocked down, but you have to help muscles regenerate, then these secondary interventions may be helpful. So reversing muscle atrophy, some of you may have been involved in the MY29 study back in the mid-2000s, and this was an antibody to myostatin. Myostatin is a regulator of muscle growth, and if you knock it down, you get increase in muscle mass and increase in muscle strength in animal models, and there are even human examples of that. And so, but as you know, the MY29 study, which was done on multiple muscular dystrophies, including FSH, did not work. And so a lot of companies have been working on basically version 2 and 3 and 4 of similar drugs, trying to have a more complete knockdown of this pathway. So this is the study that the drug company called Acceleron is about to start, and this is the ACE-029 study, and this is a locally acting myostatin inhibitor. And so this is the animal study that was done, and you can see, so one side was injected with the placebo and one side was injected with active drug, and you can easily tell which side got the active drug. The muscle is much bigger and much stronger. And studies have been done in normal individuals, and it was shown to be safe and to also cause increase in muscle volume. And the next step is to take it to what we call a phase 2 study, where the objectives are to find an optimal dose, measure drug effectiveness in 2 muscles, and the targeted muscles are muscles that are commonly affected in FSH, and these are the biceps and the tibialis anterior. The tibialis anterior is the anterior leg muscle that helps lift your foot up when you're walking, and the weakness in that muscle causes foot drop, and as a lot of you know, the biceps is, both of these were chosen because they're easy to access, but they're also critically important for function in a lot of patients with FSH. And so these seem to be kind of logical targets. And so that study is about to start, I think, within the next couple of months. I don't know exactly all of the sites that are involved. It's going to involve injections of those muscles once every 3 or 4 weeks. And again, there's going to be a dose escalation, meaning they're going to go try and see what the optimal dose is, and then go on for a longer study with an active arm and a placebo arm as well. So the other approach is trying to tackle inflammation. Inflammation is something that we see not infrequently in muscles of patients with FSH. And the company called aTyr started a study with a drug called Resolaris. And this is a medication based on a naturally occurring enzyme that plays a role in making protein, basically translating DNA from a particular gene into a protein. But and so this is kind of a new function for this enzyme that was discovered by this company, in that it not only helped translate DNA into protein, but also seemed to have some anti-inflammatory properties. And so they were interested in trying that in FSH. The study was, and so this involves weekly injections, as you heard. So again, this kind of limits the access to some patients unless they're living within driving distance from a site that's involved. And so initial studies were started, and several doses were tried, and the study has stopped now that the drug company is looking in details at the results of the studies and trying to kind of figure out what is the best way to move forward after that. Presumably, there's potentially there's a potential that the study will continue, probably starting early in 2017. So these are the two active studies that are occurring in the U.S. If you go to, other ongoing studies, if you look at clinicaltrials.gov, there's a clinical study of neuromuscular electrical stimulation on the effect of that on muscle function in patients with FSH. There's another study of creatine monohydrate in children with FSH, which is about to start, I think it's starting in middle of 2017, and this study is occurring in Australia. So future clinical trials, these are the ones that are active. Again, I think having been at the first FSH consortium meeting many, many years ago, and now I can tell you that it's a completely different landscape, even though there's not a trial that's imminent right now other than the ones I told you about, but we are all very excited because I can tell you 5 years ago there was not a single drug company that was attending this consortium, and now there's at least 10, 15 companies that are involved in FSH trials, at least working on potential interventions. And so, again, I go back to this initial figure. Again, we talked about blocking DUX4, and there are many ways of doing that, either with small molecules that can actually wrap up the chromatin where the DUX4 is and close it down. There's a CRISPR technology that you heard about earlier. There's antisense oligonucleotide, and there's RNAi, all of which are being worked on right now. And so these are still possibilities. I think, again, as we talked about, looking at some of the downstream effects it is important to look at those other aspects because you need as many tools as you can get to treat this and to have the optimal effect. And just, I know this is probably, you're too sophisticated for this, but again, this is a very good resource about finding out what's going on in FSH, and also what studies are involved and what sites are recruiting patients. Thank you.